Imagine this. A scientist walks into a lab. A mouse is sitting quietly in a tiny chamber. The scientist presses a button. A flash of blue light enters the mouse's brain through a hair-thin optical fiber, and suddenly, the mouse freezes. Press the button again. The mouse runs in circles. Press it again. The mouse stops feeling fear. No drugs, no surgery happening in real time, just light. And here's the part that sounds like science fiction. Scientists weren't just stimulating the brain, they were turning specific neurons on and off like light switches. Welcome to the strange and fascinating world of optogenetics, a technology so powerful it lets researchers control brain circuits with light. And yes, it's real science. The brain, the most complicated machine ever built. Your brain contains about 86 billion neurons. Each neuron is a tiny electrical machine. They communicate using electrical pulses and chemical signals. These pulses control your memories, your emotions, your fears, your habits, even your faith and imagination. But for most of human history, scientists faced a massive problem. We could listen to neurons, but we couldn't control them precisely. Think of it like trying to understand a symphony while only being allowed to hear the entire orchestra at once. No isolating the violin, no muting the drums, just noise. Then one day, scientists realized something extraordinary. What if neurons could be controlled with light? The discovery hidden in pond scum. The story begins in an unlikely place, algae. Tiny green organisms floating in ponds. Some algae contain special proteins called channelrhodopsins. These proteins do something magical. When light hits them, they open microscopic channels that allow ions to flow through the cell membrane. In simpler terms, light equals electrical signal. A few brilliant scientists had a wild idea. What if we could put these light-sensitive proteins into neurons? If it worked, neurons would fire whenever light touched them. That idea sounded insane, but in 2005, researchers at Stanford, including Carl Deisseroth, demonstrated it worked. They inserted the gene for channelrhodopsin into neurons. Then they shined blue light. The neurons fired instantly, just like flipping a switch. And with that experiment, optogenetics was born. The first time scientists controlled behavior. Once scientists realized neurons could be controlled with light, they did the obvious thing. They tried controlling behavior. In one famous experiment, researchers targeted neurons that trigger fear in mice. Light on, mouse froze in terror. Light off, fear vanished. Another experiment activated reward neurons. Mice began repeatedly walking to the corner of the cage where light stimulation occurred, even though there was no food. They were chasing the artificial reward signal. In other words, scientists were writing behavior directly into the brain, which raises a terrifying and fascinating question. If you can control neurons, could you control thoughts? Turning memories on and off. Now we enter the deeper rabbit hole. In 2013, researchers at MIT did something that sounded impossible. They implanted a false memory in a mouse. Here's how. Step 1. They identified neurons activated during a pleasant experience. Step 2. They tagged those neurons with optogenetic proteins. Step 3. Later, they stimulated those neurons during a mild shock. The mouse's brain rewired the memory. Now the mouse believed the pleasant environment was dangerous. A memory had been edited, not erased, not suppressed, rewritten. This experiment shook neuroscience because it suggested memories aren't stored like files. They are stored as patterns of neurons firing together. And if you can control those neurons, you can rewrite the pattern. Healing the brain with light. Now, before this starts sounding like a supervillain origin story, there's a powerful medical side. Optogenetics has helped researchers understand Parkinson's disease, depression, epilepsy, addiction, and chronic pain. For example, scientists discovered specific dopamine circuits responsible for addictive behavior. By stimulating or suppressing those circuits in animals, they could reduce compulsive drug seeking. In Parkinson's research, optogenetics helped map the exact neural pathways responsible for tremors. That knowledge is improving deep brain stimulation treatments used in humans today. And here's the most exciting part. In 2021, scientists reported early human clinical progress using optogenetic therapy to restore partial vision in a blind patient. Researchers delivered light-sensitive proteins to retinal cells, then used special goggles to send light signals to those cells. The patient began detecting objects again. A blind person seeing shapes because neurons were engineered to respond to light. The ethical rabbit hole. Now we reach the philosophical edge. If we can control neurons with light, what else becomes possible? Imagine turning off traumatic memories, removing addiction, suppressing violent impulses, enhancing learning, boosting motivation. Sounds amazing, but now imagine the darker possibilities: emotional manipulation, artificial pleasure loops, behavioral control. Optogenetics today requires genetic modification and implanted fibers, so it's not something anyone can secretly do. But the technology proves something profound. Your thoughts, your fears, your desires all exist as patterns of neurons firing inside your brain, a storm of electricity. And optogenetics proved something extraordinary. Those storms can be controlled. Right now, this technology lives mostly inside research labs, but its discoveries are rewriting neuroscience, because we're beginning to understand something ancient philosophers always wondered. What exactly is a thought? And if a thought is just neurons firing, then the next question becomes impossible to ignore. Who or what controls the switch? If this blew your mind, type "light" in the comments and subscribe, because the deeper we go into science, the more reality starts to feel like science fiction. And trust me, this rabbit hole is just getting started.